And we have two other structures on both sides. Does any people know what they are?
Can you repeat the question again?
Yeah, the question is, you can see two panels on both sides here. Here and here. Does any people have an idea what they are? Any people know what is the structure here which is going out of the cell?
That looks like the tunneling nanotube.
Yeah, here in the middle, yes. But this one here. Nobody have an idea? It is a primary cilium, and I will show you later what this primary cilium is very important in the cell. I asked this question, I just want to know what is the background of the people, where you are, and if you will follow, or I need to go more and more slow to explain what are these structures. Here are the microvilli, which are normally found on epithelial cell on the apical surface of the epithelial cell, and we will come back as well on this. So, my lab, we are studying this markers called CD133 or prominent 1. As you can see here, this molecule has 5 transmembrane domains, 2 large glycosylated loops, as you can see here. Each loop have more than 250 amino acids. They are quite big, and this is unusual for a membrane protein, a polytopic membrane protein, when you have many transmembrane domains. You have 2 small loops. The N-terminus, after the cleavage of the signal peptide, is located outside the cell, in the lumen, but the C-terminus part is located in the cytoplasm of the cell. So, my lab is interested to this molecule, but as well of the plasma membrane of the CD133 positive cell. So we are interested to understand how the plasma membrane of cell is organized. It's not like a balloon, they have different shape, and they have different subdomain in the plasma membrane. And we are studying this with this marker. The CD133 positive cell are expressed in different type of population. As I mentioned 2 weeks ago, this molecule is found in stem and progenitor cell. And that's a lot of people become very, very interested to these prominent positive cell because they can isolate stem cell. However, it's also expressed in differentiated cell. So when you isolate the stem cell, then you need to keep in mind that differentiated cell can express as well. And this cell, at least subpopulation of the CD133 positive cell, have the capacity after differentiation to come back as a stem cell. That's what they call facultative stem cell, meaning that you have a cell which is differentiated, and upon stress, it can de-differentiate it and behave like a stem cell. These CD133 positive cells come very, very important in the field because 2 independent groups have shown that this molecule can ally or can isolate and use the isolation of the cancer stem cell. The first paper, it was 2 papers by Sheila Singh in Canada, where they found that you can isolate the brain's cancer stem cell with this molecule. And Catherine O'Brien, also in Canada, both groups are in Toronto, they can show that you can do the same with a colon cancer stem cell. And because of that, then the literature about these molecules really, really go up. And if you go now in PubMed, you will find more than 6,500 papers related to CD133. As I mentioned, this molecule become more and more important, and also the cell express it, and we can find this in many organs, and that's why this molecule become very famous. Indeed, all these organs express CD133 positive cell. In my lab and with the collaboration, we are focusing on brain, retina, blood, kidney, where these cells are expressed, the CD133. But we also make paper with almost all other organs that we have it. So as I say, this molecule become very, very famous, and the cell which express it as well. So what do we know? It turned out that prominent-1 turned out to be the first member of a new family of pentospan membrane glycoprotein, meaning with 5 transmembrane domains. In our case, we found this molecule in 1995 as a marker of the neuroepithelial cell. As you can see here, these cells, and as you know, these cells will generate all neuron and glial cells that we have in the brain. And CD133 was identified with an antibody. It's called 13A4. It was in this case in the mouse. And this molecule was found specifically, as you can see here in green, on the apical surface of this neuroepithelial cell. Here on the lateral, you have the lateral side and the basal side. 1 month after we published this paper, a group in US, a group of David Buck, have found the same molecule but in human. And in this case, it was a marker of the hematopoietic stem cell. So the homology between the two proteins, the mouse and the human, was quite different. They have only 60% of amino acid identity. And in the beginning, we are not sure if they are the same molecule or a related molecule. It turned out to be the same. So the sequence, primary sequence of these two proteins are quite different. They share only 60% of amino acid identity, but the structure is exactly the same. So after we cloned, a few years later, we found another molecule we call prominent-2, and this we found in mouse and in human. This molecule is in the structure identical to prominent-1, but they share only 30% of amino acid identity. Now they are quite different from the primary sequence, but the organization is exactly the same. And I will make just a short story to say that prominent-2 behaves exactly like prominent-1, except it's not found only in the apical surface of the cell, but it's also found in the lateral surface of the cell. That's the major difference between the two proteins. And we found this molecule in mouse and human, also in different species. And we found a third one, what we call prominent-3, in lower vertebrae, like here, the axolotl, and as well in the zebrafish. You probably know that these two models are used for regeneration, and it turned out that these 3 molecules are expressed there and can be upregulated when we make amputation of some part of these animals. So highlighting again that they can play a role in stem cell. It's also expressed in Drosophila, and I will give a talk in 2 weeks just before Marius Adai about the retina, and I will show you the role of prominent-1 in the retina. In addition to prominent-2 and prominent-3, we also found more than 20 different splice variants of prominent-1. And many of these splice variants have different C-terminus, meaning that they probably have different interacting partners. So as I mentioned in the beginning, this molecule became very famous because it's expressed by the somatic stem cell, meaning adult stem cell. And we found in neural, and the people in California found in hematopoietic system, but it's also a stem cell in the kidney, prostate, pancreas, and many tissue. This molecule can highlight the stem cell. However, you need to be aware that it's also expressed in differentiated cells, like the epithelial cell, photoreceptor, and glial cell. And indeed, we cloned this molecule on the mouse kidney. And then the mouse kidney is highly expressed, as you can see here, on the apical surface of the epithelial cell. Since you have a lot of prominent there, now we use a library which was built from this tissue, and we isolate the clone, and then we found the protein. It's also expressed in photoreceptor cell, and I will come back in 2 weeks on that, and as well in glial cell. For instance, it's highly expressed in astrocyte, and as you know, the astrocyte can play also a role as a stem cell. And here you have the myelin, which are produced by the oligodendrocyte. As you know, the myelin, it's a lipid membrane that will go around an axon, and it will make protect the axon, and this molecule is highly expressed in the myelin. So we use different systems, as you can see here, and indeed, what we found in one system, like epithelial cell, where this molecule is expressed on the apical surface, we look if it's also true in other systems, like the hematopoietic stem cell. If you remember last week, I was talking that prominent one is found in the uropod of these cells, or in photoreceptor cell, where you have the outer segment. The outer segment of photoreceptor cell is very important for the vision. Okay, you will have all opsin, rhodopsin molecules that will be located in this structure. And prominent one is found at the base of this structure. And this structure here is very important, this protrusion here, to make the new photoreceptor exist. And of course, we also use the cancer stem cell to see how these membrane of these cells are organized. So what's very striking with this molecule that it's found in the plasma membrane, but it's not everywhere there. It's concentrated in some structure that protrude from the flat area. And these structures are from, for instance, the microvilli, the primary cilium, or other like filopodia. When you have a migrating fibroblast, they will make at the front a lot of filopodia, and this molecule is highly concentrated there. Are you familiar with this structure, microvilli, primary cilium, filopodia, or it's the first time you listen to that?
I'm familiar, for example.
Okay. So what will be the function of a microvilli, for instance?
In the intestine is the absorption part. And
yes.
For example, in the nose also, it's like a filter, usually, for microparticles.
Yes, but what it's doing in general? Like if you go in intestinal cell, what these structures are doing?
You mean like actively doing, or?
Yes.
I didn't get the question.
Even passively, what they are doing. If you go in column, we call the surface where are the microvilli is called branch border. Because you have a lot of microvilli all around the surface. And it turned out that these increase the surface. And why they increase the surface of the membrane? Because you have a lot of transporter and an enzyme which allow the digestion of everything coming there, cut in small pieces, and then they are uptake by the cell by transport or even by endocytosis. Normally with transport protein. That's why it's increase the surface. You talk about the primary cilium, which you have one per cell, then you will find a lot of signaling pathway, protein-involved signal pathway are located there. And upon ligand, the binding of the ligand of a given receptor, you can have a cascade of events afterward coming inside the cell. So normally, the microvilli increase the surface of the cell. Primary cilium play a role in signaling, for instance. Okay. So this molecule turned out to be highly expressed in the protrusion. As you can see here, that's the neuroepithelial progenitor, which I was talking in the beginning. And I was showing the green staining was located here on the surface, the apical surface. This surface is different than the lateral one because you will have in the beginning tight junction, which allowed to seal the two surfaces differently. Now the composition of the protein here is different than the lateral one. And at the base here, you will have the basal membrane, and all these cells will generate the new neuron that we have in the brain. And this is called the lumen because this is inside, I think, the cerebral fluid. So you can have also material which are released by the cell, and they will go in the fluid here. So when we look by fluorescence, I see in the beginning, I show you in the beginning, you have a very strong staining on the apical surface here. Now, if we go more deep, to localize a protein, meaning by electron microscopy, what we found, as you can see here, by immunoglobulin labeling with the antibody against prominent 1, this is in this case we are on the mouse, you can see the labeling only in protrusion. These are microvilli-like structure. You don't see any staining on the flat area. It's only in the protrusion. Here, it's a primary cilium, a developing primary cilium. As you can see, very, very strong staining on the structure and almost no staining on the surface of the cell. Highly concentrated there. So when we transfect cell, the first cell we transfect was called NDCK cell. It's an epithelial cell line, and you can see here the apical surface of these cells. Here you will have the tight junction, which allows the separation between the apical and the lateral part of the cell. And again, you can see the staining of prominent 1 or CD123, very, very strong in microvilli. No staining on the flat area. It's the same with another intestinal cell line. You can see, yes, strong staining again, and very often on the tip of the microvilli. And I will come back to this later. And more strikingly, when we transect fibroblasts, even overexpression, as you can see here, this molecule really loves plasma membrane protrusion. Here you have a very small microvillus-like structure, and you can see here the very strong labeling and absolutely nothing on the flat area. Now, this molecule really, really like to go in plasma membrane protrusion. Because of that, we call this molecule prominent, from the Latin word prominere, to stand out, to be prominent. And it become prominent 1 because then we found another molecule, which we call prominent 2 afterwards. So within the plasma membrane protrusion, we found that this molecule is found in plasma membrane microdomain, as we call, or lipid raft. And any people, I was talking 2 weeks ago about the lipid raft, and any people know a little bit more about that?
So basically, lipid raft is like a densely organized region within the cell membrane where there are closely packed lipid molecules and embedded proteins in it.
Yeah. So as you mentioned, the lipid are specific subdomain of the membrane. As you can see here, they are rich in specific protein, notably the protein which are linked with a lipid like a GPR anchor, but you have also transmembrane protein there. And these domains, as you can see in green, are highly enriched in cholesterol and also in sphingolipid. You can also find ganglioside like GM1, GM3, they will define different type of membrane microdomain or lipid raft. So these domain or platform can play a role in signal transduction, intracellular trafficking. Indeed, these platforms are very important for targeting protein to the apical surface of an epithelial cell. Now, the lipid raft will form at the level of the Golgi. They will put a specific vesicle and they will go to the apical surface. And consequently, they can play also a role in the polarity of the plasma membrane by making segregation of different type of protein. Any idea how can we study the lipid raft? Or the main technique that people are using to identify if a protein, a given protein, is found in lipid raft?
I'm not sure how we can identify a specific protein, but can C-Loudron spectroscopy be used to check if it's a lipid draft or not? Is that particular part of
By microscopy, you mean?
C-Loudron spectroscopy
Okay.
or C-Loudron microscopy? I mean, it's spectroscopy actually, yes.
Okay. Don't forget they are very, very small, yeah? They are small and they are very dynamic. And that's a lot of people who are confused with in this field because they can form very rapidly and dissolve properly. But you have one biochemical technique that people use to identify the lipid wealth. And any of you will continue to work in the lab in the future, you will probably come to that. And as I was mentioning, one of my colleagues just few meters from him discovered this field or pushed this field, which is called Kaisimon. And you have some very specific technique to use it, which raises also a lot of questions. Any people have an idea? If I mean detergent, does that mean something for all of you? If you do immunofluorescence, for instance, and you want to label a protein which is found inside the cell, in cytoplasm or Golgi, what are you doing? Does any of you have done immunofluorescence microscopy? Nobody?
No.
So nobody have the chance so far to label a cell for microscopy to observe by immunofluorescence.
I've done microscopy, but not with a cell, with molecules, with purified components.
Okay, but what the people are doing microscopy, if you want to do fluorescence, what you do normally? You have your cell, and then?
Maybe we use an antigen or an antibody which is labeled to a specific fluorophore.
Yes.
Which will bind to the cell surface or a particular
Exactly.
organelle.
Yes, that's the first step. If you want to label a protein like prominent one on the surface of the cell, first you can fix the cell, and then you come with your antibody, and then you have secondary antibody which is coupled to a fluorochrome. We'll label the cell, and then you can go to the microscopy to look. Now, if the protein that you want to see in your cell is inside the cell, it's not on the surface, what you will do?
We can't fix the cell, that means?
Yeah, you can fix the cell, and then what you do, the antibody can go inside the cell.
Only if it is permeable through the membrane? Cell membrane?
Yes, but your antigen, it's not on the surface, it's inside the cell.
Is it possible to genetically modify the cell so our target molecule will be fused with fluorescent protein?
Yeah, but this is quite complicated, no?
Yes.
So what the people are doing when you do, if you have your cell here, you want to bind, but you want that the antibody will go inside.
Maybe add something that increases the fluidity of the membrane.
Yes, and how it is called?
Detergent.
Yes, detergent, exactly. So what the detergent are doing?
It will partially disassemble membrane.
Yeah, but how?
I think it is amphiphilic. if I am correct.
Yeah, they will permeabilize the membrane. That will allow the antibody to, they will not dissolve completely, but they can dissolve some part, and the antibody can then go inside the cell, find the epitope or the protein that you want to label, and then you will label the, you never have any cost about that labeling of mitochondria, ER, Golgi structure?
No.
Not at all.
No.
Okay. So normally that you have your antibody, you fix the cell, you permeabilize, meaning you will make some kind of hole in the cell, and then your antibody will go inside the cell and will bind, and then you can do your fluorescence coming with secondary antibody in the same way. Then your structure that you would like to see it with a given protein will be illuminated, and then you can observe by immunofluorescence microscopy. All of this to come to a detergent. Now, the way that people identify the lipid raft, they claim that the material found in this structure here are insoluble in some given detergent. And the major detergent that people are using is called Triton X100. Any people have listened to this name of the detergent before?
No.
No. Okay. That's a general, or some people have listened?
Maybe general.
Yeah, okay. It's called, the detergent is called Triton X100. Okay. And what this detergent is doing, it can solubilize the whole membrane except this structure here, the lipid raft. But if you want to know if your protein is found in lipid raft, you just take your cell, you solubilize them in the Triton X100, and then you just do centrifugation. And if your protein is found in lipid raft, it will just become insoluble and it will pellet in your fraction. Now you can have the fraction afterward insoluble and insoluble material, and you just run a Western blot, and then you can see if your protein is found in this structure. So we have done this with CD133 or prominent 1. I have done that. And I use, of course, the detergent called Triton X100. And I found out that it turned out to be totally soluble, meaning that prominent will not be associated with lipid raft, which are insoluble in Triton X100. However, I decided to use other detergent, and one of them is called the Lubrol WX, different structure. And it turned out that this prominent one can be found in membrane microdomain, which are insoluble in Lubrol WX. And I will come back to this in a few seconds. So the lipid raft play a role here in different functions, signal transduction, intracellular trafficking, plasma membrane polarity. So prominent one is found in lipid raft, but not the classical one, which are insoluble in Triton X100, but those who are insoluble in Lubrol WX. So I mentioned here that prominent one bind to the cholesterol. And we have found that using a compound, it's called the photocholesterol. The compound was developed by my colleague in Heidelberg. And this compound here is very similar to the cholesterol, but you have a tritium group here. It's radioactive. And you have a carbon residue here, which after UV light, this compound becomes very, very, very active. So every molecule very, very close to the photocholesterol will bind to it. So we do this experiment. We incubate our cell with this compound. We put the UV light, and then afterward we solubilize. As you can see here, after immunoprecipitation of prominent 1, this molecule binds very, very, very strongly to the cholesterol. You can see here below the ER form, when prominent is still inside the cell, do not bind to the cholesterol. It's highly specific for the plasma membrane protein. And this is quite important because the membrane microdomain here are dependent of the cholesterol. So meaning that if we remove the cholesterol, the molecule that we have identified becomes soluble in the detergent that we are using. So this is quite important to show that this molecule here, it's a cholesterol binding protein. It may organize also some membrane microdomain. So then we look by fluorescence. If this molecule is found in membrane microdomain which are lubrol resistant, it may differ for the protein which are normally found in the triton X100 resistant membrane complex. And one of these molecules which is found in triton X100 is called the placental alkaline phosphatase, which is a GP anchor protein. So we found that this molecule is found in triton X100, but not prominent 1, which is on the other hand found in lubrol resistant membrane complex. And if these are not artifacts, we should be able to see in vivo what's happened to these two proteins. So we label the cell with antibody against either prominent 1 or the placental alkaline phosphatase here. And what we can observe that both proteins are not together. So they are going in different type of membrane microdomain and also structure. As you can see here, I will make a short story. It turned out that the alkaline phosphatase is found in the flat area, and prominent 1 is found in protrusion structure like the microvilli and the primary cilia. So the biochemical data that we observe can be reflected with the morphological data, that the two proteins are not together. And more strikingly, afterward we look for lipid. As I said, there are membrane microdomain which are dependent of lipid. And we found out that prominent 1 is highly enriched in the lipid, what the lipid is called GM1. I mentioned two weeks ago this lipid. And they are together in the microvilli. The GM3, if you remember, I was showing in hematopoietic cell where you have prominent on one side and you have the GM3 on the other side, as you can see here. They are totally separated. The GM3 will be found with the placental alkaline phosphatase protein, while the GM1 will be found to be together with prominent one in microvilli. The primary cilium, the situation was more complex because we can find indeed both gangliosides, GM1 and GM3. And I can see you here, show you here an example of staining. You have prominent 1 here, and you can see a very good colocalization on the surface of the epithelial cell with GM1, which is not the case when you label with CD133 and the ganglioside GM3. As you can see here, they are separated. So you can have on the surface not only different type of subdomain, microvilli, primary cilium, or any other type of protrusion, but you can have also within this surface different type of membrane microdomain. In other words, you can have different type of lipid raft on the surface of the cell. So that's a small cartoon which summarizes this part where we show that prominent 1 is specific for plasma membrane protrusion, microvilli here, and these structures are soluble in triton X100, but insoluble in lubrol WX. And there, prominent interact directly with the membrane cholesterol. On the flat area, you will have protein like GP anchor protein, or one of them I show you was the placental alkaline phosphatase, and these surface will be insoluble in triton X100. And this experiment is quite important, and you can see we found this long time ago, 20 years ago, but we are still publishing paper on that. Why it's so important? Because, as I show you here, prominent was soluble in triton X100, and one of the main techniques when people do fluorescence microscopy, they permeabilize the cell. And to permeabilize the cell, the main detergent is used is triton X100. So when you do that, you have your cell, you fix, you put your detergent, and then you label the cell. In the case of CD133, which is really tricky, that even if the cells are fixed, if you permeabilize your cell with this detergent, you can totally extract this molecule. And the consequence of that is big, because most of people, what they are doing for fluorescence or microscopy, and also for flow cytometry, cell sorting, flow cytometry. So they will normally permeabilize the tissue with Triton X100. So given that CD133 or prominent 1 is totally soluble even after fixation, they will conclude when they take a cell type after depolymerization that they are negative for this marker. Unfortunately, and it happened many times, that this cell was indeed positive, but by polymerization with the Triton exponent, they just extract the protein. And the consequence is big, particularly when you talk about the cancer cell, do are prominent positive or prominent negative. Most of the studies have been done so far, they are done after polymerization. So the whole conclusion that you can see in the paper, prominent negative, prominent positive, can be wrong. And that's why you have a lot of contradictory in the field, that some people say, oh, no, they are positive. The other people say, no, they are negative. It's just by not chance, but not in the opposite of chance, that the people who claim they are negative are using the Triton X100. But this is just a normal detergent that people are normally using. So you need to keep in mind that when you do your staining in the future, when you will start to do that, that the detergent can extract your protein of interest. So your conclusion will be totally wrong. So keep in mind that if you don't see something, that's not mean it's not there. Okay? Does any other people have an idea which can other pitfall, I would say, of staining? You don't see anything, but indeed your protein is there. Does any people have an idea when this can occur? Your supervisor asks you, does this protein is expressed in the cell that we are using? So you do your staining and then you see nothing, but then you conclude it's not there. How an antibody binds to a protein? Where it binds? Everywhere or something? Yeah, if somebody epitope. Yes. What does it mean? Cell, antibody come, bind, here's the epitope. Okay. What's happened, if you see it, you are happy, I see it. What's happened if you don't see the staining? You have no staining.
Can you repeat the question, please?
Question is, if you do a staining, immunofluorescence staining, you have protein 6. You have your antibody. Normally, you will bind, you do immunofluorescence, then you have a fluorochrome. You can see the protein, you can see the cell is labeled, you are very happy. Your protein is there, your antibody is there, but you don't see the fluorescence. Of course, as I mentioned before, if you have a detergent like the Triton X100, you can extract it, then it's not there any longer, then you will not see it. But imagine the protein is not extracted, you put your antibody, but you don't see it. Then you conclude, oh, it's not there. But the question is, what can block the binding?
For example, protein can be unfolded or not in native state and epitope is not exposed.
That means that you need to look when you will buy an antibody, you need to look what is the epitope. It's a conformational epitope, like you say, it's depending on structure, which can change if you denature the protein, or it can be a linear epitope, just bind directly. Yeah?
Yeah.
And if it's a linear protein, that a linear epitope, you have a big chance to see it, even after denaturation. Yeah, that's one thing that you need to consider when you buy an antibody. You look where is located the epitope and what we know about the epitope. So I all time suggest the people in the lab, before you buy, look which antibody people are using in publication. And if you want it's working very nice, then you buy it. And not the opposite. Don't go to the catalog of the company to find it. Find a publication before. Yeah? Now imagine that your epitope is linear and it can bind. Even after the denaturation of the protein, the antibody can bind. But what can happen? You will not see your protein.
Maybe the fluorophore has bleached out already.
Imagine everything is perfect. Your antibody is perfect.
Are we talking about living cell, right?
No, you can fix the cell as well. You can fix the cell as well.
Something else may be bound to the target protein and epitope is blocked.
Yeah, it's very close to that. The antibody do not have access to the epitope. Often the protein in the cell, they are not alone, but they are in a complex with other protein together. And meaning that another protein can mask the epitope of your protein of interest. Now, if you come with your antibody, you will not see it because they are just masked. And this is quite important when you do staining, that when it's negative, you can see that under these conditions, I cannot see it. But that's not mean that your protein is not there. And that's why it's quite often important to use different epitope. Antibody can directly a different epitope of your protein, which can give you different type of result, Depending if one epitope is masked and the other one is not masked, then you will have a completely different result from negative to the positive. So just keep in mind when you do the staining, when it's positive, you're happy. When it is negative, of course, you will say, oh, I'm not happy and I didn't find it, but that's not mean the protein is not there. Then you need to try different detergent, you need different antibody before to really conclude that the protein is not there, okay? Just inside that, and one day I'm pretty sure you will face issue like that. And also when you buy antibody, as I mentioned, don't go to the catalog directly, go to the application, find paper where antibody will be used, and then you buy this antibody and not go to the catalog, because very, very often, particularly with this protein, many antibodies that they sell are not working at all. Just keep in mind that. So I show you that this molecule is found in the plasma membrane of cell and in protrusion. It's found in membrane microdomain. And something else, and I mentioned this 2 weeks ago. This molecule can be also found in small extracellular membrane vesicles, which are released outside the cell. So we can find this molecule in many fluids like urine, tear, similar fluids, saliva, and association with very, very small membrane vesicles. And you remember 2 weeks ago, I was talking about that. So I will come now how we found this very, very small membrane vesicle. Here you have the neuroepithelial cell, as you can see here, where in green is located CD133 or prominent 1. That's an epithelial cell, basolateral, lateral, basal membrane, and it's located only on the apical surface. Here you have the cerebrospinal fluid that you can take in the spinal cord. That's the fluid that you will have also found in the brain. And I was talking about these cells can differentiate, and they will generate, as you can see here, a neuron. The other cell here, which will be identical to the mother cell, meaning the stem cell. So I showed you 2 weeks ago that these cells can go in cell division, and they will slightly divide the cell, that one cell will not get this apical surface, and when they are losing CD133, they start to differentiate. But we found something quite interesting, that when these cells start to go in the process of differentiation, it's not only about the symmetric or asymmetric cell division can happen, but something else. It's the release of very, very small vesicles, as you can see here, CD123 containing membrane vesicles. And this occurs only when the cells are going in the process of differentiation, which is quite important in the concept of the stem cell. And they release in the cerebral fluid these very, very, very small molecules, vesicles here, which contain this one. We can see here by immunogold labeling. Okay, as you can see here, very small vesicle, less than 100 nanometer. And you can see they have CD133. That's another example here, CD133 vesicle here. And you have another type of vesicle, it's called exosome. And I was mentioning, I will mention again in a few slides again, but if you remember what I was talking about, the hematopoietic cell, they can release prominent one in association with exosome. Here's another marker of exosome, CD63, and then you can see in this case here, 133 do not co-localize or is not together with the 63, meaning that these vesicles here are not exosome, but are nonetheless released in the fluid, outside the cell. And to make a very, very long story short, we could show that these vesicles are indeed budding from the microvilli. If you remember in the beginning, I was telling you that CD133 is found in microvilli, very often concentrated on the tips. And what's happened here, when they start to be concentrated, they make a phase separation with the rest of the microvilli. They are lipid raft here. And then you have two types of membrane, like oil and water. They don't like to each other, and one will bond. And that's why we are releasing here this small vesicle, which contains CD133 or prominent one. And again here, it will be insoluble in Lubral WX. And prominent one, we have shown it also can bind the cholesterol. That they are releasing small vesicles which are enriched in lipid raft. So I was mentioning that these are dependent of the cholesterol, these lipid rafts. So when we remove the cholesterol, and you will come to the literature very often with a compound that's called methyl beta cyclodextrin. It's a compound we extract the cholesterol from the membrane. So when we incubate the cell with this, what we observe that the microvilli become like a purling microvilli, and we have a release, massive release of small vesicle, and the microvilli disappear. I think I have a picture, yes, as you can see here, the purling of the microvilli, and then you release. Okay. This is quite interesting that in this cell type, prominent is budding from the microvilli. And again, if you remember with the hematopoietic cell, I was saying that prominent one is released with exosome. And this occurs when the cells start to differentiate. It's exactly the same here with the epithelial cell, in this case with the CACO2 cell, which can differentiate in culture. And the differentiation occurs normally around day 7. And you can see at day 7 to 8 here, we have release of this small membrane vesicle. So the stem cell or the cell which differentiate release this vesicle. They can bud from the microvilli, but also from the other structure we call the primary cilium. And you can see here example where CD133 become highly concentrated on the tip of the primary cilium and the bud, and they release this very small vesicle. So they can come either from microvilli or the primary cilium. So we do not know what is the function of this very, very small vesicle, but what is interesting, we can use it as a biomarker. I show you here several examples about this CD133 membrane vesicle, which are upregulated in different diseases, like in glioblastoma, or also in the multiple sclerosis or partial epilepsy, we have observed an upregulation of these particles and the cerebrospinal fluid. So they can use, we don't know the function yet, but at least we can use as a biomarker. And interesting in cancer, depending on the grade of the cancer, we can have an upregulation or a downregulation of this particle. So all time keep in mind what you are doing in the bench can all time find an application afterward in the medical field. And that's why it's quite important to try from your basic research how this can be applied to the medicine. And of course, we can see you have a lot of people, then you need to make collaboration with your colleague in the working in hospital and so on to get sample and so on. But all time try to, what you do in the basic research, try to find an application. And this is quite interesting and it's quite exciting as well. So we characterize more. We also found that prominent one in the case of salivary gland, it's upregulated, and we have also showed that this molecule in the saliva can be ubiquitinated. So 2 weeks ago, I was talking about this cell, the hematopoietic stem cell, and I was all time saying today in the beginning of the talk, what we found in one cell type, like epithelial, that is also true with other cell type. And just to make a short story, I mentioned this 2 weeks ago, we used the hematopoietic stem cell, and we have, as you can see here, mesenchymal stromal cell, hematopoietic stem cell growing on the top, and using this, and again, CD133 is found on the tips of the protrusion on these cells. If you remember, these cells, the migrating one, as you can see here, are highly elongated. You have a structure, it's called uropod, highly enriched in GM1, where also CD133. This is very, very similar to the microvilli, which I just showed you a few seconds ago. But on the other hand, the GM3, the ganglion GM3, it's not totally the opposite. It's exactly, again, like we have an epithelial cell, where you have prominent on the microvilli and GM3 on the flat area. And then you can see here the structure of the cell when I migrate. I will come back in a few minutes about the structure. And if you remember, we showed that the hematopoietic cell, which is in contrast to the epithelial cell, we have two pools of CD133, one on the surface and also one inside the cell, which is in contrast with the epithelial cell, which is only in the epithelial surface. The pool inside is found in multivesicular body, if you remember that. This big structure will fuse with the plasma membrane and release a small vesicle, which here are called exosomes after the release. These vesicles are CD133 positive, but in this case, they will be also positive for the other marker which I was talking before. It's called CD63. And then you can see here, and they release after the differentiation, but we induce here with the 4-ball ester, they release in the extracellular milieu. So you will have two different pathways for releasing the CD133 particle. One, what we show with hematopoietic cell, that means the CD133 will go in endocytosis, early endosome, late endosome, which are also called multivesicular body, and they will be found inside the structure, as you can see here. The other marker was mentioned is called CD63, will be also found in this vesicle. And this large structure can fuse with the plasma membrane and then release the small particles, which are called exosomes. This can also occur in cancer cells. And now, just to show you, if we go back to the epithelial cell here, you have two ways that they can, prominent can be released of CD133, can bud directly here from the microvilli and release outside, or they can bud from the primary cilium and release as microvesicle or ectosome. Okay, so the people in the future who will work with extracellular membrane vesicle, you should think that they can be different type or different nature of these vesicles. They can be budding directly from the plasma membrane or released after the fusion of the multivesicular body with the plasma membrane. Okay, and they're all together, and it's make a soup. And indeed, the situation can be more complex, and nobody really, they are just starting to think about that. When you have a vesicle, they can have different origin. Okay. And it's quite important because in cancer, you can have both, which are together. And again, this is important to know because many people are looking at these vesicles as a biomarker of a given disease. So they release this particle during the differentiation. And we propose in this context that the stem cell, or even the cancer stem cell, when they go in the process of differentiation, they will release this small membrane vesicle which contains CD123. As I say, we can use this particle as a biomarker, but the exact function we don't know. We know that they can also be, if you remember 2 weeks ago, they can be uptaken by the mesenchymal stromal cell, making a communication between one cell type and another cell type here. But they can way to communicate. But we propose that specific lipid raft from stem cell or plasma stem cell can have the protein or any other kind of determinant that can maintain the stem cell property of the cell. So when they lose this membrane microdomain, they go in the process of differentiation. Also in the future, it will be very, very interesting to know which other protein, which other lipid can be associated with prominent 1, and if they can play a role in maintaining the property of the stem cell. So if I summarize this part here, so the plasma membrane of the epithelial and non-epithelial cell contain distinct type of membrane microdomain, and their prominent interact with the membrane cholesterol and associate with the cholesterol GM1 containing membrane microdomain found in the plasma membrane protrusion. Prominent 1 is released in various body fluids, like here, saliva, urine, similar fluid, and this is quite interesting because they can be used for monitoring cancer. If you have a cancer in the kidney, the level of particles which are released in the body, in the urine, can change. So you can monitor some cancer. The same here with the seminal fluid. So it can be very, very important to use as a biomarker, although we don't know the exact function of these vesicles. And we have shown that the neural and the hematopoietic stem and progenitor cell release the prominent positive extracellular vesicle during the process of differentiation. So this is quite important because if we want to amplify the pool of stem cell, and this is quite very important if you go into regenerative therapy, you need a lot of stem cells. As I said 2 weeks ago, they are very, very rare cells. So if you want to increase the pool of stem cell, one way might be to block the release of these vesicles. So if they are not releasing this vesicle, maybe they will still like a more stem cell, and then you can increase your pool of stem cell. Now, if you go in cancer, you would like, particularly with the cancer stem cell, if you would like, not necessarily to kill, but to eliminate this cancer stem cell, which proliferate and get more and more cancer cells, if you want to eliminate them in the way, you can push, maybe you can try to find the mechanism of pushing the release of this prominent extracellular vesicle, prominent extracellular vesicle, which will push the differentiation of these cells. And when they will be differentiated, they will not proliferate any longer. So it's maybe 1 or 2 ways to use an application playing with this small membrane vesicle. So before we go, do we take 5 minutes off? It's fine for everybody? Do we take 5 minutes, a break of 5 minutes? Or we
Yes.
Sorry, yes?
Yes.
Okay. So 5, 6 minutes, fine? Yeah, okay, I see, good. Good? So, any people have an idea about what can be the function of this molecule, CD133? If we summarize, this molecule is found in the plasma membrane. It's highly concentrated in plasma membrane protrusion, like microvilli, primary cilium, filopodia. Within this membrane protrusion, this molecule is found in membrane microdomain, lipid wafts, where they can organize these lipid wafts by binding to the cholesterol. So from these observations, what can you conclude about the function of this protein?
Maybe cell-to-cell communication?
Sorry, mutation?
Maybe cell-to-cell communication?
Yes, but what a molecule can do by himself?
I don't know, signal transduction.
Signal transduction? Yes. Normally, signal transduction, we are thinking about receptor. You have a receptor bind to the, you have a ligand binding to the receptor, and then the receptor will start a cascade of events to the cytoplasm. So far, you have no ligand, natural ligand have been found for this protein. In other words, you have no evidence it is a receptor. And as I mentioned in the beginning, it's expressed in many, many different cell types, which are often really different to one to the other. If you take photoreceptor cell versus epithelial cell or even hematopoietic cell, they are really, really different cell types.
Maybe it is involved in the cell motility?
If I go You mean cell motility?
Yes.
Yeah, they can play a role, but what's happened there? What it will do? What this molecule will do in the primary cilium or in microvilli?
But change the shape of the membrane?
Yeah. Any other people have an idea? So we will go more in deep on that now. Okay, so you can see the structure of CD133, the 5 transmembrane domain, and terminus located outside the cell. The C terminus, as you can see here, you have different exons, and we show it here, and I call facultative exon because in many splice variants, you can remove some of them and you have different type of C terminus, meaning they can bind to many different proteins there. You have here a splice variant, which is a very, very small exon, which around 7 amino acids, which is located in the N-terminal part. We don't know the function of this exon. Indeed, we don't know the function of any of these exons. So two things here are quite interesting for us. So two groups. First, a group in Quebec, Canada, Montreal have showed that prominent 1 can be phosphorylated and can be phosphorylated on tyrosine 828. And then a group in US could have shown that this phosphorylation is very important for the interaction between prominent 1 and an enzyme. It's called PI3 kinase, phosphoinositide 3 kinase, as you can see here. This enzyme is 2 subunit, and the phosphorylation of CD133 is very important to bind to the subunit called P85. And this interaction here between prominent 1 and PI3 kinase allow the transformation of lipid in the inner leaflet of the membrane, as you can see here. Indeed, this enzyme will transform a lipid, it's called PIP2, you have the name here, into a PIP3. And this is quite important in signal transduction indirectly because many proteins like HARC are pathway will specifically bound to the PIP3 lipid. So prominent here, the interaction with PI3 kinase can transform the lipid in the inner leaflet of the membrane from PIP2 to PIP3. Then another group here, also in Toronto, the group of Mark here, has shown that this lysine here can interact with an enzyme. It's called histone desacetylase 6 here. And what this enzyme is doing is transforming, removing the acetylated group of tubulin. And I will come back to this. And now you have also here a group in France have showed that prominent 1 have the binding site for a lipid, which is called GM1. And now this makes all sense because I show you, and we have shown before, that prominent all time co-localized with this lipid in plasma membrane protrusion. So to go more deep about the function of these domains, we make mutation. So we have mutated the natural binding site, GM1 binding site in prominent 1. We changed 2 amino acids. And in this mutant that we call 2M, 2 mutation, we analyze what's happened with the cell. So you have the N-terminus. Here, the small exon, which is 7 amino acid, is present only in S2. Then we make the mutation in both S1 and S2 mutant. And then we express in cell. And we use again the NDCK cell, the cell which I showed you in the beginning, that prominent 1 is going in microvilli. So when we express the wild-type protein or the mutant, what we found that first with the wild-type protein, we increase the number of microvilli. So when we put this overexpress this cell, we come with more protrusion on the surface. And when we express the mutant, we even increase more the protrusion on the surface. Meaning that prominent 1 can probably play a role in the formation of this structure or the stabilization of this structure. You can see here the increase of this. Not only the number of microvilli increase, but also cluster of microvilli. And this is true when we do the wild-type protein and also the mutant by comparison to the non-transfected cell. We increase the number of microvilli and the cluster of microvilli, which is quite interesting. But now when we start to look more in detail how they look, the microvilli, after overexpression, we come with quite a big surprise. As you can see here, a small microvilli in normal condition, as you can see here, single, going straight forward, as you can see here on the map. When we do overexpress the white type molecule, white type prominent one, what we found, something striking. You can see here that the microvilli shape completely changed. We have like a V shape. It's not only one microvilli, but you can see here by scanning EM, you have many microvilli make a double structure, which is quite unusual. Or even like microvilli look like fused together, as you can see here by scanning EM. Even more strikingly, the mutant, where we make 2-point mutation in the site where prominent 1 should bind to the ganglioside GM1, we observe at least 3 different types of shape. One with a very big head, meaning that the microvilli membrane is detaching the cytoskeleton inside, which in this case for microvilli are actin, and they are detaching. And I will come to explain to you why. Or we come with a shape which is very similar to the curling shape when we are removing the cholesterol. If you remember in the beginning of the talk, when we remove the cholesterol, we have a purling shape of the microvilli, and this is very similar. Or another one structure was quite very interesting. You can see, and this like a frozen vesicle which normally will bud, but it's not budding any longer. So we can see the structure very often, which we normally never see in the normal condition. Now, this induces different type of mutant, which may explain the function. So you can see here the ratio, normal short microvilli, normal microvilli, what we have in MDCK or an MDCK cell transfected only with a vector. But when we start to put the white type prominent, either S1 or S2 splice variant, we can start to have branched microvilli. And this is even increasing with the mutant when the branched microvilli really increase. And we also come with microvilli which have this shape where they start to detach. Any people know how is the structure for microvilli? You have the membrane, you have actin inside, and how this all connected to each other. No? So we can go a little bit more deep about that. You can see here, normal microvilli expressing prominent 1 or not expressing prominent 1. As you can see the structure here, inside the microvilli, you have actin. It's actin-based structure model. And this actin is very important because you will have protein like myosin here. You have different class of myosin, which will link the actin to the plasma membrane. So they are like a decoration and they are allowed to make this structure like a tube here. So then when we have a prominent there, what we observe, that prominent start to be concentrated on the tip and can bud as a vesicle. That's what we have in normal situation. Now, when we overexpress prominent 1 or even the mutant, we observe 2 major phenomena. The branched microvilli or more microvilli cluster that can generate it. And this can be generated by the action of protein and enzyme like the wave that will bind to the actin cytoskeleton, and then you have a complex, it's called ARP2-3 complex here, which will bind to this wave protein. And what these proteins together, they will initiate the formation of a branched actin network. So they are doing branching actin. And they're all time doing this in a ratio of 70 degrees as we will have here. And this branching of actin is very important normally when you have migrating fibroblasts. You will do the lamellipodia in the front of the cell, and then you have a branching which made this lamellipodia, this actin structure there. And that's what we observe here with the mutant of prominent 1, or the overexpression of prominent 1, will create this branching-like structure, meaning that maybe prominent have something to do with these protein here. In the case of where we have the feeling that the membrane becomes bigger while swollen, can be related to the conversion of a lipid that's called PIP2 to the PIP3. As I said in the beginning, the enzyme PI3 kinase can play a role here by converting these two lipids. These lipids here are very important to bind to myosin, not only PIP2. So if myosin binds to PIP2 in the inner leaflet, myosin binds to the actin cytoskeleton, it's allowed to the microvilli to keep its shape. If PI3 kinase, by binding to prominent 1, will transform PIP2 and PIP3, then the myosin molecule will not bind any longer to the membrane, and then the membrane will swollen. And that's the model that we have. We have proposed that prominent 1 within the lipid raft, or by binding GM1, we allow to keep the proper structure. If we block or we interfere with that, interaction can occur and transform the shape of the microvilli. So what we do, if it's true, if the binding of prominent 1 is important to keep the shape normally, and when we create mutant, we change the shape. That means if I go back here, if we change the shape here, we will have an action of PI3 kinase, what will transform PIP2 and PIP3, and then this detaching. So to evaluate that, we make the mutation in the mutant, which already have the mutation in. We make an additional mutation where the phosphorylation of prominent can be blocked. So if we block the phosphorylation of prominent, it will not interact with PI3 kinase, and the PIP2 will not be transformed in PIP3. If it's true, this idea, then we should not affect the microvilli shape. All through, we will have mutation here that do it. And it's turned out to be the case, as I can show you here. When we do the mutation, we had the mutation in the tyrosine residue, 828 or 819, depending on which size variant, we highly reduce the effect of the first two mutations. For instance, 70% of cells have this shape. When we create this mutant here, we go to 5 or 2% in the case of the splice variant, have it. So we really block the transformation. So by mutating GM1, this will affect the phosphorylation of prominent and then its interaction with PI3 kinase, creating this effect. So if we use drug that will block either the PI3 kinase or the ALK2/ALK3 complex, we completely block here the formation of this weird branched microvilli or the microvilli with a big head. Now the drug completely blocks this, meaning that the interaction of prominent with this protein here, or maybe this one, can affect the microvilli structure. And to evaluate that, we just do immunoprecipitation. As you can see here, the wild-type prominent, the one which have two mutations in the GM1 binding site, and the one which will have the mutation in addition of the phosphorylation site in prominent, which normally is important for binding to PI3 kinase. And as you can see here, when we do immunoprecipitation and we analyze by immunoblood, you can see in the wild-type situation, we can immunoprecipitate P85 of PI3 kinase, but with the mutant, we increase this binding. And when we make the mutation of the phosphorylation, which is important to bind to PI3 kinase, we block it. And the same is true with the ALK2/ALK3 complex. As you can see here, mutation in GM1 site stimulate this interaction, and now it's blocked by addition to other mutation here, which go to PI3 kinase. So altogether, it's a protein complex formed between prominent and between enzyme, which modulate the actin cytoskeleton and the membrane as well, in case of PI3 kinase. Now, if we go in the knockout of prominent 1, what we observe is a bit more complicated. We observe microvilli. These are microvilli in the kidney, which instead to be a normal shape, as you can see here, start to be fused. So we have a disorganization of the microvilli structure upon knockout of prominent one. Now they really do something in organization of plasma membrane protein, as you can see here. And this is quite interesting, not only for microvilli, but if you do to other cell type. Remember, I was showing in hematopoietic cell, prominence is highly enriched in the uropod. What does it mean if it's enriched there? What you will find there? Any idea? We know that prominence is there. We know that GM1 is there. And what else will be there? It's written here on the slide, indeed, microvilli. As you can see here, the structure here in the uropod will be enriched in microvilli. It's exactly what you have here. Now keep in mind that prominence found in plasma membrane protrusion, each time we see on the surface staining of prominence, we can imagine it's full of microvilli or any other protrusion. As you can see here, that's exactly the case. The uropod structure is not a smooth surface like here, but it's plenty of plasma membrane protrusion, as you can see here. So what we do, we have different examples here of the uropod. What we have done, we knock down the expression of prominent 1 in the hematopoietic cell. As you can see here, using small interference RNA, we pull down its expression. And then we look what's happened on the structure. And surprisingly, what we found, and we can quantify that the uropod structure have almost no microvilli any longer after silencing CD133. If we silence another molecule, which is also found in microvilli, it's called CD82, no effect is observed. And it's really, really specific for CD133. Now, it affects the structure of microvilli not only in epithelial cell, but as well in the non-epithelial cell, in this case, in hematopoietic stem cell. And the same is all true if we now move to fibroblasts, where we overexpress prominent 1 to compare to the cell which do not express, we can generate a lot of philopodia, as you can see here. This philopodia change after the mutation of the GM1 ganglioside and change again when we make the mutation either with the phosphorylation site or the phosphorylation site alone. Then we change really the structure of the plasma membrane protrusion also in fibroblasts. And this is quite interesting because fibroblasts, as you know, the philopodia in fibroblasts, but also in cancer, is very important for the migration, metastase, if you think about the cancer cell. So now, how all these proteins can all link together, but it's known that one protein is called IQ-GAP1, can make a large complex between PI3 kinase and HOP2/3 complex. So we propose that maybe prominent 1 can also be part of this large complex. And if we do IP, or immunoisolation, we can pull down this molecule, which is highly more increasing when we make the mutation in the ganglioside, GM1 ganglioside site here. This is quite interesting because normally these proteins are involved in formation of the lamellipodia, where you have the branching actin here. And this is quite important for the migration of fibroblasts, but also cancer cells. As you can see here, two examples, when the people put down IQ-GAP1, they can block the migration or the evasion, in this case, of the ovarian carcinoma cell. So you have an active reorganization of the membrane here in front of migrating cell. And probably in the cancer cell, prominent can play also a role there. That was quite important. And in the future, what we would like to study is how all these proteins here interact more in detail to each other. And it's also known that if you knock down prominent cancer cell, the migration will go down. So this slide can summarize what we we see from the beginning of the second part. And this is quite interesting because here you have the plasma membrane, you have the outer leaflet of the plasma membrane, the inner leaflet. The interaction of prominent 1 with the GM1 ganglioside here can favor its phosphorylation on the cytoplasmic side. So you have a crosstalk between the outer leaflet to the inner leaflet. And this phosphorylation will stimulate this interaction with PI3 kinase. And as you know, PI3 kinase will change the lipid on the inner leaflet from PIP2 to the PIP2. Such interaction, we can modify the structure of microvilli, but can also modify the structure here of a structure called the primary cilium. As you can see here, it's a primary cilium, and I showed you in the beginning that prominent 1 can bud from the primary cilium. And if you see here, when we create this mutation, we have formation of very, very funny network on the tip of the primary cilium. And this is quite interesting, and we are now actually working on that. So what people know about the primary cilium? I was asking this question in the beginning. And that's why it's quite important to know what is going on there, because it's a center of signaling in the cell. And as you can see here, you have many pathways which are directly linked to the primary cilium, like sonic hedgehog, the Wnt pathway. Probably many people have listened this before. You have many pathways, or the notch signaling pathway, are all linked to the cilium, where you will have receptor of these ligand that will be stimulate effect in the cell. And the first step is the ligand will bind to the primary cilium. The complex with the ligand can move down and go inside the cell and make action. And many, many diseases, as you can see here, will be affected when many can be created when the primary cilium will be affected. That is quite important to keep in mind. Yes, it's really a center. And you have now a lot of paper coming every month about the structure, the interaction of protein within the primary cilium. And I'm pretty sure that some of you will probably work in the near future on this. So just example here, what the primary cilium can do. The primary possess very specific composition, lipid and protein. I show you in the beginning, they can have GM1, they can have GM3 together in the same structure, while the microvilli have only GM1. And what they do, they convey extracellular signals to control the cellular process during the development and the homeostasis. And signaling pathway like Wnt, sonic hedgehog here, transforming growth factor, bone morphologic protein, receptor tyrosine kinase are all located in this structure. And many, many diseases called idiopathy can be caused by the effect on protein involved in the formation of this structure. So they are very, very important structure. So what can be the impact of prominent 1 in the organization of the structure? As I said, two major pathways are associated, hedgehog and the Wnt pathway. And they all depending of the structure because the receptor are located there. And when the ligand is binding, something happens down. Either the receptor is moving or a signal is sent down. Like in this case, in the case of sonic, proteins called gli will move to the nucleus and then change the expression pattern of the cell. I showed you in the beginning, CD133 is found in primary cilium, budding from the tip, and this is also done during the differentiation process. More the cell goes differentiation, more they will release it, which is quite important, meaning the action of the cilium may change during the process of differentiation. So now we go back what prominent can do, and I show you here that this enzyme, histone desensitisation, can play an important role there, which is bind to prominent. What this enzyme is doing, this enzyme, one major difference between cilium and the microvilli is the structure, cytoskeleton structure. The microvilli are based on actin. The primary cilium are based on tubulin. Okay, they will do similar array, but it's a tubulin. And the tubulin will go together in the case of the primary cilium when they have a modification. It's called acetylation. When the acetylated tubulin go, they will form the primary cilium. They will go and form. This enzyme, what it's doing, indeed, it's removing the acetylated group of tubulin. And as the result, the cilium disaggregates. It's falling down. Okay? So when this enzyme is free, it removes the acetylated group, and then the cilium is going down. So what we do to study that, we make mutation in the binding site of the eastern deacylase 6. We change the lysine. And now what's happened? First, we overexpress prominent 1, and we observe that here, as you can see here, 2 examples, or by fluorescence here, prominent molecule, acetylated in red. What you can see, we increase the length of the cilium. When we do with the microvilli, overexpression of prominent, we create more microvilli. But in the case of cilium, we don't create more cilium. We still have 1 cilium, but they have become longer, which is quite striking. Now, when we express the mutant here, which will bind to the eastern deacylase 6, what we observe, the cilium are coming shorter. You can see here by fluorescence or by scanning EM. Very, very short. We can quantify that. As you can see, we have defined the cilium in 3 lengths, longer than 5 micrometer, between 3 and 5 micrometer, or shorter than 3 micrometer. This is MDCK cell non-transfected. When we transfect with the normal prominent, we highly increase many of them, as you can see here, the length of the cilium. They are coming longer. When now we express the mutant, we totally lost this capacity to make longer cilium. And not only we lost this capacity to make longer cilium, but we have more and more cells without cilium. Some smaller, and we lost many cells don't have cilium any longer. So what happened? So here you have a staining with the acetylated tubulin. Normal situation, overexpression of prominent, you seem to have more acetylated tubulin coming. But now with the mutant, the acetylated tubulin is going down, which is meaning that the enzyme, which is binding to prominent, will not bind any longer, will be free, and will remove the acetylated tubulin, the acetylated group of tubulin, and then you will have the cilium coming down. It's exactly what's happening here. And what we can observe, on a very rare long cilium left over, we can see again a purling effect many times, meaning that this vesicle here will be released and the cilium will come shorter and shorter. And as a consequence of the expression of the mutant, we can detect more vesicle in the medium, meaning that cilium comes shorter and we release more vesicle. So that's one way. Prominent will interact with this acetylated tubulin. The enzyme removes the acetylated tubulin and will allow to make the cilium. When we bind, it's not binding longer. The acetylated tubulin will be removed from the cilium and they will resolve. So that's one way, putting down the cilium formation. But now how can we explain that prominent without mutation can increase the cilium, the length of the cilium? One protein which is involved in this is called RL13B. It's a small GTPase, and they have been shown that people, when you overexpress this protein, the cilium are coming longer. And to make a very, very long story very short, we say maybe prominent can also bind to this RL13B. So we do immunoisolation of this enzyme, this protein here. And what we can see, we can pull down prominent 1. We do not pull down the mutant, which is the mutant which should interact with the other enzyme, which remove the acetylated group, and they don't pull down here. If we do immunoisolation of prominent 1, again, with the wild-type prominent 1, we can immunoisolate this IL-13B, but not with the mutant. So we have proposed here that prominent can play a role in the formation of the ciliary compartment, either by interacting with IL-13B, making long cilium, or by interacting with the histone 6, which can reduce the cilium. So you have a balance between these two structures. And as you know, the cilium are very important also for cell division. When the cells start to divide and so on, the ciliary length is changing. Okay? So this is very important because if you don't have this, then you don't have a normal proliferation or differentiation of the cell. As you can see here, in the mutant prominent here, when we overexpress it, the proliferation of the cell, at least in cell culture, is affected. So the mutation affects the proliferation, which is directly linked to the assembly or disassembly of the primary cilia. This is in vivo. What to do with stem cell? So to reproduce what we see in vivo, we completely switch the model. And we use a model of the teeth of the mice. The incisor, the two big teeth in the front, which, as you know, they're all time growing. And doing that is meaning that you have a pool of stem cells which continuously renew and will generate the new teeth. And in this context here, it's called the cervical loop of the epithelial stem cell niche in the mouth. You have a pool of stem cells here, which will go in the process of proliferation. And then you have the cell which is called the TAC, transient amplifying cell. These will proliferate, and then they will go in the process of differentiation, and they will form the amyloblast, which are the cells which create the enamel of your teeth, and so on, dentin, and so on. Now you have here a beautiful system, a linear system, which allows you to see what will be the impact of a protein in the formation of stem cell, proliferation, differentiation. As you can see here, you have indeed, oops, come back here, you have indeed the expression of prominent 1 in the stem cell, but also in the transient amplifying cell, as you can see here. And which was very, very interesting with this system, that the primary cilia have a different shape. They are very long in the stem cell here, and more the stem cells start to amplify, which is linked also to the cell proliferation, they are shorter. They have more variation because the cell is dividing. And as I showed you before, the cilium is linked to that. And you can see here, more the cells are dividing, more you have a switch lower, larger, lower, which is not the case in stem cells when they are not dividing. So since prominent is expressed in both, maybe quite interesting to know what will be the impact there. First, we can see that in the stem cell, to the transient amplification pool, you have the expression of IL-13B, which is going down, as you can see in red here. Long microvilli, you have a lot of IL-13B. And more you go for the transient amplifying cell, the amount of IL-13B is going down, and also the size of the cilium, which is exactly the opposite with the other enzyme here, the histone 6. In the stem cell, you have very little about these protein, and more the cell is playing a role in the primary cell play a role in coming shorter, longer, you have an increase of this enzyme. So from the stem cell to the transient amplifying cell, you have two proteins, which one is involved in primary cilium formation, and one was involved in the disassembly of the primary cilia. And we propose that in this case, that prominent 1, by binding these two proteins, can play a role in formation and disassembly of the primary cilia. More IL-13B here, you make the assembly. More Histone 6 here, disassembly, and prominent can play a role there. So if we now measure the length of the cilia in the wild-type animal, you can see here the mean length of the cilium change drastically between the stem cell and the transient amplifying cell. It's going down, which is normal because you have more transition here, cell division. And then when we analyze the knockout of prominent 1, what we observe that this transition do not exist any longer. Okay, so the cell division will be highly affected, and we don't see too much a big difference between the two states. So really prominent play a role by regulating the formation and the disassembly of the primary cilia. As a consequence of that, in the knockout of prominent 1, where we can see here that the NML will be highly affected. Indeed, the function of the differentiated cell, the amyloblast here, is not working properly. And we are also shown then in culture by taking these cells, the primary cell from the wild type and the prominent knockout, that they are not answering any longer to the sonic HR pathway. Now we really affect the organization of the primary cilia. So that's a cartoon which summarizes this part here, that in stem cell, we have a long cilium prominently located there and can answer to the sonic hedgehog. When they go in the transient amplification, when you have activation of the stem cell, the cilium, as I show you, can vary in length. And what's happened, the cilium are coming shorter. And I will not go too short to it, but a very important transcription factor associated with sonic hedgehog will move, and will move indeed together with prominent 1 in the nucleus of the stem cell. So that you can, the transient amplified stem cell here. So you can see here that you have a transition of 2 proteins, and prominent 1 can play a role in this transition, which is quite exciting in the field of the stem cell and the organization of the stem cell. So this is true for the cilium found in the stem cell, but the cilium are found many other places as well. And you have a disease, it's called diabetes, and Anthony Gavalas will come in December to give a talk. And just to summarize quickly this disease, you have 2 types. The type 1 is when the immune system attack and will destroy the cell are producing in the pancreas the insulin. And you have a type 2 diabetes when you can produce insulin, but you will not have any action any longer. So the cell become non-responsible to insulin any longer. So we come to prominent in this field. That was a collaboration we have with people in Argentina, and they are interested to know what and how the diabetes can affect the brain. Just to summarize you what you have in the brain here, normal brain here. What you will have the ependymal cell, and you have one type of quiescent stem cell, which indeed is an astrocyte, and you have a primary cilium here. And this astrocyte can be activated, it's prominent CD1 positive, and can generate neuroblasts, can generate new neurons. Okay, so you have a primary cilium here in this astrocyte, or it's called B1 cell, but you have also cilium, a lot of cilium that's called multicilium in the ependymal cell, as you can see here. They form like a wheel model where you have the stem cell in the middle, and you have this ependymal cell all around. Both cilium, the primary one, and the multicilium also contain CD133. And these cilium here are very important for the movement of the cerebrospinal fluid that you have in the brain. So they are moving. And with this, you have the movement of the fluid. And this movement can also affect the migration of the neuron. And this is when we talk about the mice here, it's very important that you can have movement from the neuron that they have in the brain to the nodes. Okay, that's quite a nice, very nice system to study that. And to see if prominent can have an impact on this structure in the diabetes, what we create 2 models, type 1 diabetes or type 2, and these are created by 2 drugs, as you can see here. If we put only one drug alone, the streptozotocin here, we will create a model where we have a type 1 diabetes. Now, the cell producing insulin will be destroyed. When we combine with the second drug, we will create a type 2 diabetes model, as you can see here. And what we observe in the brain, this is a subventricular zone, what we can observe, you can see here the staining of prominent 1 in the cilium. Nestin is a marker of the neural cell, as you can see here below. Okay. So when we go in the type 1, we have not a big difference. Prominent is still found in the cilium, as you can see here. But surprisingly, in the type 2, the cilium look quite different, as you can see here and here. We totally lost nestin as a marker of the stem cell here. And if you look carefully on this structure, this is the multicilium found in the epidermal cell. You can see quite a disorganization. Here they are quite uniform and pointing in the same direction, but yet they go other direction. And if you look here with high power view, normal situation, type 1, you can clearly see a flow in one direction, this cilium. Now in the type 2, they are going everywhere, as you can see here, or they even collapse together. This is quite interesting. Nobody have showed that before that the type 2, you can have problem in the brain. We are working with CD133. We know that CD123 is expressed on these cilia. So what happened? You can see here in normal situation, and you can see on the tips, the staining of CD123 in green here. You can see very, very well the label all around the cilia. Now, if you go in the type 2, indeed the type 1 do exactly the same. Now, if you go in the type 2, as you can see here, you totally lost. You don't see any longer the staining of CD133. Indeed, it's collapsing more at the base of the cilium, you know? Now, you don't have in the tip, but they are moving down. Cilium are still there, and as a result of that, the cilium tend to collapse to each other. As you can see here, normal situation, all the cilium are going up, very separate to each other, but now in the knockout of prominent 1, the cilium have tendency to stick to each other on the tips, but as well all along them. So they are going, instead to be like a flower, they are collapsing to each other, as you can see here in this case. Totally. And this is quite interesting, meaning that in this case, prominent might play a role there by maintaining the proper, not only the structure, but the proper distance between each cilium. So please keep in mind this for the talk we will have in 2 weeks with the photoreceptor cell. So here we have showed that prominent 1 modified the architecture of the plasma membrane protrusion, notably the microvilli and the cilium in the epithelial cell, and the filopodia and lamellipodia in non-epithelial cells. Prominent 1 interact with PI3 kinase, ARP2/3 complex, and IQ-GAP1, which could have an impact on cell migration in non-epithelial cells. Prominent 1 interact with the histone deacetylase 6 and IL-13B and regulate the length of cilium, and hence the function of the cilium-dependent signaling pathway. And together with other protein partner, prominent may have anti-adhesive property, allowing the proper organization on the biological membrane. So it's not involved only to make them, but also to keep them in a normal situation and not collapsing. So what we found with all of this by studying only a single protein. So we found a lot of things with the cell biology of stem cell. For instance, we have shown that the stem cell, when they are going in the process of differentiation, they can release very small membrane particles in the extracellular milieu, meaning that they can release the property of the stem cell, which we can found together in a membrane microdomain or membrane lipid raft, which be together and prominent, and they release them. They release by 2 different mechanisms, either by budding from microvilli on the primary cilium in epithelial cell, or in a non-epithelial cell like the hematopoietic stem cell, they release as exosomes after the fusion of the multivesicular body with the plasma membrane. In cancer, we may have both pathways together. 2 weeks ago, I was also showing that prominent 1 can be exchanged from one cell to the other one by this very tiny tube. It's called tunneling nanotube. That's another way to exchange prominent 1 to one cell to the other one. Or the cell division, symmetric and asymmetric cell division, can play a role of to eliminate or to share CD133 or the lipid membrane microdomain associated with prominent 1. So we have also showed this was true for the epithelial cell. We also show for the non-epithelial cell. We also show that the biology of the epithelial cell have quite some remarkable feature there. That prominent have showed that they can play a role in formation of microvilli and budding of vesicle from the microvilli, but as well from the primary cilium. So what can be interesting with this molecule? So prominent 1 is used as a cell surface antigen for the immunoisolation of stem cell and as well cancer stem cell. So it's already quite huge impact in the field. The prominent stem cell can be used for cell replacement. Indeed, it can be used for the bone marrow. We can immunoisolate the hematopoietic stem cell based on prominent, and to go for bone marrow transplantation. It's also a lot of paper about heart. They can also inject the bone marrow-derived hematopoietic cell, and they can help the heart after an attack to recovery. They can play maybe also a role in the kidney, or more importantly, spinal cord injury. So, that's more a question. We are talking about replacement based on prominent stem cell, but maybe the vesicle containing prominent 1 can be also important for the stem cell transplantation. And we know very little about that. It's totally a field which needs to be explored. And we can have other possibility to use therapeutic perspective related to prominent 1 and prominent 2. I did not mention too much thing because we know very little about prominent 2. We can use to as a biomarker, monitor the tissue engineering cell replacement. If your tissue is not working any longer, like the kidney you replace, then you will expect that if everything goes well, the new kidney that you will get after transplantation will start to release this vesicle, and you can find in the urine. So they can be used to monitor the new tissue. Of course, they can use also to monitor disease, as we have shown with the glioma. You can monitor disease by following prominent 1 or the particle containing prominent 1. It can be used as a molecular target for cancer. We know that the overexpression of prominent can create cancer. Maybe we can think to target the cancer cell by targeting CD133. But nonetheless, in this case, we need to keep in mind that prominent 1 is not expressed only in cancer cell, but also in normal tissue. So if you want to target them, you need to be very carefully, because otherwise you can kill normal cell, particular those in the retina, which express prominent 1. We need to be nonetheless careful, but it can be used eventually with combination with other marker. And it can maybe used as well for gene therapy. I did not mention anything today. I will do in 2 weeks with retinal degeneration, where you have mutation in prominent, which can cause a retinal disease. And we can go more in that direction. So I have the chance a few years ago to make a book where I asked many colleagues all around the world to make a chapter. We have 15 chapters in this book, and each chapter is talking about different tissue and organs. So as you can see, we can touch almost every part of the body, of the human or the mice body, with this single molecule. And I'm pretty sure that if people will go more deep with many other proteins, they can do exactly the same. Good. More question? I will put, after the talk now, I will put this presentation in this OPAL system so everybody will have it. And you can. Any question? So in conclusion, we say that prominent 1 can play a role in organization of plasma membrane protrusion. Microvilli, primary cilium, filopodia, lamellipodia. And since this touch almost every cell type in the body, so this molecule plays quite a big role there. Question? Nobody have any question? So what do you think you need to know for the examination at the end of all of this? No question at all? So how's it going in general? You're fine with all the presentation with the doing by Zoom or BBB by internet?
Yes, but is there a chance that we have access to the slides maybe one day before the lecture so we can look at them and then it is easier to understand the context of lecture?
Okay, this was complicated for you today or not in general?
Like some things were completely new for me.
Okay.
For example, about different membrane protrusions, I haven't studied this before.
Yeah.
Yeah. And also, yeah, I wanted to ask what should we focus on for the exam?
That's what I was asking before, what you would think will be important for the examination here.
Maybe function of prominent one and
I will not go too much. No, I will not go too much in detail. I will be more general.
okay, but what like function and mechanism?
Not really, no. I will be more general about stem cell. What are the stem cell? What they can do? What are the properties of stem cell? How can you isolate stem cell? So it will be quite general. I will not go in detail to ask you which mutation on prominent one can do what and one. Don't go. None of the questions will go in this type of direction. They will be general question. The only thing we go in more in detail is to give you an overview what we can do with a single molecule and how far we can go. But I will not go in this direction for the examination. It will be more simple.
Okay, thank you.
More question?
I have a question.
Yes, just quickly, just reading the comment. Yes, I have my colleague for the anatomy, Joseph Yes, I will give a talk 2 weeks ago, and I'm just waiting for his reply to get the lecture. But don't worry also, I will not come to question too much on the lecture of other people unless the one that you already have with the mesenchymal and the hematopoietic cell. So people should not stress with that. Okay. So beside the stem cell, people asking me which other cell express CD133, it's almost many cells express CD133. Almost all epithelial cells express CD133. It's also expressed in the retina. And as I showed you in the beginning, glial cell like oligodendrocyte, as you know, the oligodendrocyte will make myelin around the axon, they express prominent. Indeed, myelin, it's also plasma membrane protrusion. Astrocyte also express prominent ones. So many cell types indeed express it. That's why you need to be very carefully. If you want to target stem cell which express prominent one, you need to be very careful that other normal cell express it. So if you come with an antibody which has something to kill a cell attached to it, you may kill normal cell. That's why people need to think about that. So far, yes. Somebody asked if prominent is found only in protrusion. If they are found in the plasma membrane, most of the time, about 99% is found in protrusion. Okay, and that's why the name comes. That we are pretty lucky when we put the name, but we look carefully before. It's all time found in plasma membrane protrusion. So if you have a staining on the cell, now you can think that this region where you have the staining, you will have some kind of protrusion there. And people which are stressed with the examination, it's a 20-minute examination. I have question there. My colleague in the past was Martin Bonozer coming to this lecture, to the examination. And technically, if you know a little bit of base, you can done in 5 minutes. Okay, that's not very difficult. We go up to 20 minutes because we try to help as well people. So I can do an examination with written examination, but if you don't know the answer, then you are catch. The advantage of the oral examination, we can help you really to come to the answer by changing the way that the question that will help you to answer. It's really more easy, I think, than the written examination. Good? So next week, and you will have talk until the end of the day, but as you say, I can all time come if you have many questions to me. We can all time set up another appointment with this BBB blue button stuff. We can all time do it after a talk or before, like if you really feel that you have question, we can do another talk, or people can come, we can have a meeting. We see in my office, I all time keep the window open. It's quite cold here, but nonetheless, if people have any question, I'm all time available for all of you.
But also, I wanted to ask, is it possible that we have slides before the lecture?
Yes, I will ask my colleague if they are willing to send before or not. Mine is not a problem to put there. The only thing I can put is not a problem. It's just because I like to ask questions, but I think it's quite difficult for all of you to open, close, and answering this question. That's why if I put before, then you have all the answer of the question. So, but I can answer my colleague as well. It's not a problem.
Thank you.
Good. Fine. So, thank you all of you, and we talk to each other. I think it's in 2 weeks. I think it's the 4th of December. Just I will do just before my colleague Marius Ada, and I will inform them if they can upload the talk before. That would be nice. Thank you. Bye-bye.
You are currently the only person in this conference.